I'm Jessie. I'm the content strategist here at RadiaSoft, and we're going to get started with our webinar in just a couple of minutes. Before we get started, I would like to remind you to please submit your questions using the Q&A button at the bottom of your screen rather than the chat. That helps us keep track of all the questions, and we will address them at the end of the webinar. We're expecting to run about 20 to 25 minutes today, and then we'll have our Q&A session after that. If you need to leave, don't worry, we'll send you the recording about a week out from this initial live webinar session. So today's talk is called Applying Computer Vision to the Automation of Sample Alignment in Neutron Beamlines, and will be presented by RadiaSoft President, Dr. Jonathan Edelen. We also have research scientist, Dr. Abdu Diao with us today. So Dr. Edelen earned his PhD in accelerator physics from Colorado State University. After that, he continued on and was selected for the prestigious Bardeen Fellowship at Fermilab. While he was at Fermilab, he worked on RF systems and thermionic cathode sources at the Advanced Photon Source. Currently, John's focused on building advanced control algorithms for particle accelerators, including solutions involving machine learning. So we're glad you're here. Welcome to the RadiaSoft webinar series. And if you're ready, John, go ahead and take it away.
Thanks, Jessie, for the introduction, and thanks everybody for joining us today. So today I'm going to talk about a fairly specific topic of applying computer vision to the automation of sample alignment in neutron beamlines. And essentially, we're talking about how to take a fairly common tool in computer vision and why that's useful for neutron beamlines and how we can apply it to neutron beamlines to improve the operator experience and the user experience. So a little bit of background about neutron beamlines and why they're important. Usually in the accelerator community, when we think about users, we focus a lot on X-ray science and a lot on X-ray light sources because tends to be more X-ray users. There's a few more X-ray light sources, but neutron sources are actually quite important for basic science, useful for various scattering experiments to do magnetic analysis, as well as just understanding various crystalline structures and understanding how complicated materials interact with neutrons. So neutron scattering is an important and a growing field of science. There's currently 8 neutron sources being operated in the US. You're probably familiar with some of the larger ones. For example, the 2 at Oak Ridge National Lab, the spallation neutron source, which is driven by a high-power particle accelerator, and then the high-flux neutron reactor, which is a reactor neutron source, also operated at Oak Ridge. So that's sort of the epicenter of neutron science in the US. But then there's, of course, NIST and Los Alamos have neutron sources, and then there's a couple of other smaller neutron sources scattered around. Worldwide, there's around 18 operational neutron science facilities. I linked there's a list of them here if you're interested. And perhaps more interestingly is that there's 57 sort of total active, inactive, and planned neutron science facilities worldwide. So when you start to dig into it, neutron science facilities, while you may not always hear about them, there's definitely a lot of neutron science going on and a lot of facilities that support this kind of research around the world. One of the challenges with neutron science specifically is that it's very important to align the sample inside the beam. And this can be challenging, and also it can be time consuming. So we're focused here on 2 different specific beam lines. There's the Topaz single crystal diffractometer and the HB2A powder diffractometer. And they work with 2 different methods for aligning the sample. For the Topaz beamline, the sample is aligned via an optical camera. And for the HB2A beamline, the sample is aligned with a neutron camera. So for the neutron camera, what you see on the right here is an example of the sample. That's this can here. And then this is what the operator will see is basically a shadow of the sample in the neutron beam. And then for the optical camera, you see a picture of the mount, and then you see a picture of the sample. And these samples can be somewhat amorphous, and sometimes they're large, sometimes they're small. And it can be challenging to pick them out in an image and to align the beam. Alignment's important because the experiments tend to be at different temperatures. And when you vary temperatures, it can induce motion in the sample, and that motion needs to be compensated for. And if you have motion in the sample when you're collecting the data, what'll happen is that you'll finish the experiment and the data will be collected and they won't be able to reconstruct the crystalline structure or the structure of the sample. So it's very important that you're able to compensate for temperature changes in the sample environment. Also, the samples, especially at topaz, they can be rotated. And when you rotate the samples to image it at different angles or different incidences of the beam, that can cause a precession in the motor or a precession in the sample position just because the sample has a weird shape. So it's important that you can automatically compensate for these types of motion in the beam when you're operating the machine, not only for just speeding up the operation of the experiment, but for ensuring better data collection. And also when you want to do remote operations, if you want to have users around the world operating experiments 24 hours a day. So it's pretty important to try to streamline this process to improve the overall environment for scientists at the beamline. So why is this a computer vision problem? I mean, it's pretty clearly a computer vision problem because you have images in both cases. There's an image, either an optical image, which is a very straightforward computer vision problem, or you have a neutron camera, which generates an image, which is also basically a computer vision problem. And here we're interested in object identification or really image segmentation. So can we draw a contour around the image and then use that contour to identify the center of mass of the sample or to just position the sample or identify if there's multiple samples in the beam, which I'll show an example of that a little bit later. Computer vision is a fairly broad field. Typically, the simplest thing in computer vision is just image classification. So this is the case where you have an image and you say, this image is a cat or this image is a dog. And that's just a very simple classification technique. You could use convolutional neural networks for that. And that gives you a very straightforward, that's a very straightforward problem. And then sort of the next step in computer vision is image segmentation. And this is where you get into contouring or edge detection. And that's often used in science. It's used in the medical community a lot. Image contouring is a very well-known application of machine learning for segmentation of like MRIs or segmentation of other medical images. So it's a well-known and sort of common application for computer vision in the medical side. And then image fusion, of course, is when you have different imaging modalities and you want to bring them together into one image and try to merge that into a single high resolution image. So you have like an MRI and a CT scan, for example, and you want to bring them together into one and then say, this is the high resolution or high fidelity image that we've generated from these sort of different imaging modalities. So like I said, for sample alignment, in the case of the two different beam lines, we have a sample mount, or in some cases, we just always see is the shadow of the beam. So on the top case, I'm showing what the operator would see is the image of the sample or an image of the sample mount in the environment, and then there's a crosshairs for the beam. And the crosshairs allow you to do what we have implemented right now, which is a point and click alignment. So the operator can say, hey, I recognize this as the sample. They can click on it, and then the sample will move into the center of the beam crosshairs, and then the sample will be aligned with the beam. What we want to do is basically automate this process by automatically contouring the sample using training data, and then automatically adjusting the position of the sample to center it within the beam. For the HB2A example, where we have a neutron camera, it's a little bit more complicated. The sample is not always easy to see. In this case, it is pretty easy to see the shadow of the sample, but you can see on the lower right-hand corner that there's another secondary shadow in the image, and that's actually a secondary sample because they can change which samples they're viewing at any given moment. And the ability to quickly identify, hey, there's the main sample and there's a secondary sample in the beam, that's important to being able to make sure that you're not polluting your data with information, scattering information from two different samples at the same time. You want to only be illuminating one sample at a time. So it's important from an object identification standpoint to be able to say, hey, there's two samples here, or there's one sample here, and also be able to align the sample with the beam in an optimal fashion. So let's talk about the UNet a little bit. The UNet is the sort of most widely used, I would say, class of encoder-decoder network for image contouring or for computer vision as sort of a basic starting point. It's a pretty broad class of neural network. It's an encoder-decoder network, so it's not like an autoencoder. An autoencoder where you have the training data, the input data and the output data are the same. In the encoder-decoder network, it works in a similar fashion where you're reducing the complexity down to some feature space or some latent space, and then you're increasing the complexity again for reconstruction. Although now you have input data and you have target data. So here, usually the encoder is used to systematically reduce the feature complexity while also increasing the number of allowable features. And then the decoder basically resamples and up converts the image, the data back into an image. So you can start with an image of a fixed dimension and you can stop with an image of fixed dimension. And that's kind of the basics of a UNet. And next, I'm going to get into some examples. So here's just, I highlighted a few examples here. If you Google, UNet implementation in Keras, you'll get a lot of different examples. There's pretty good literature available on this. And I'm going to just take a little bit of time to step through the example for this webinar, which is the one on just keras.io. And I've made some changes to it, but that's like the basic example that I want to step through. Okay, so the nice thing about this example is that the data set is freely available and it's just easy to pull from the internet. And it's a great place to get started because it's already contoured and you can play around with the different hyperparameters or play around with the different image parameters and understand, how to build a UNet for a particular application. So this is just the data loading code here. And again, I'll include the link here. And everybody that participates in the webinar should get a copy of this. So you'll be able to modify the Jupyter Notebook as you see for your application or just play around with it. It doesn't use a lot of external libraries. All the image loading and processing is done with matplotlib, and then TensorFlow and Keras, of course, for the machine learning part of it. And I personally like to use scikit-learn for train test split just because they have good tools for that. A number of examples out there will create this stuff from scratch, totally fine. But for this example, I've modified it to include the scikit-learn version. So the example, now we're in this block here, the example loads the images from a file tree. And here's where you can specify the image sizes, the number of, and the batch size. The original example had an image size of 160 by 160. I was trying to play around with speeding it up a bit for the CPU that I'm running on. If you're running on a GPU, of course, you have more flexibility. But if we're running on a CPU, I just was downsizing it. So you can, if you want to just downsize it to speed it up and do quick iterations, you can just change the image size here, and that basically crops the image. And then you can also adjust the batch size here. And then this is just finding the paths for all the data so that it can be loaded into Keras for training. And this class basically allows you to reference the paths and then bundle that up into a data set that can be used for training. And then so this is the instantiation of the scikit-learn based training and test split. So this is basically taking, you're getting X train, X val, Y train, Y val. These are the inputs and outputs, and they're basically the image paths. So you're doing the training and validation split based on the image paths, and then building up the image data set from the paths. So you don't have to actually load all the images in, which is fairly convenient because that can be kind of intensive to do that. And here is just a simple visualization showing this is the input image, we have a cat, and then this is the output image, which is a contoured cat. And again, this is kind of like you have one contour that you're trying to target. In some cases, you have multiple contours, and those get a little bit more complicated. But for the simplest computer vision case, you say, I just want to contour this object, and this is what you're looking at. So then this is the next function basically goes through and defines the model. And the model starts off by defining the input dimensions. So the input dimensions based on the image shape, and then the initial entry layers, and then the encoder layers here. So here you have the encoder network, which is defined based on the number of filters. So if you wanted to, I did 64, 128. I think the original example went to 256. So you can play around with these numbers, and you can say, oh, I want to have 3 layers of encoder, or I want to have 6 layers of encoder with different dimensions. And it's pretty easy to just go in and modify them. But each layer has the convolutional, the batch normalization, has the pooling. So that happens at each layer. There's multiple steps. And then it steps down to where you have more filters, and it does the same operations in sequence. And then on the decoder side, you're basically reconstructing the image complexity while reducing the number of filters. So now we start at 128 and step down to 64 and then to 32 as we reconstruct that image into the output. So the second part is the decoder. And then this function just allows you could create the model by specifying the image size and the number of classes that you have for that model. So you could run multiple of these in the same notebook, or you could just run one. The training is fairly straightforward. So specify a loss function, specify the optimizer. I'm not going to get into the details of that for this particular example because I wanted to focus on sort of the UNet is the tool, and there's lots of different ways in which you can use a UNet for this type of problem. And I think that based on some of these resources, you can sort of go figure out what makes most sense for your application. And then I won't run the training, but this was just training for 20 epochs. You can visualize the output. So this next command just basically gets the data for the reconstruction, and then we can just plot it as an image. And here we're looking at the predicted mass, the target mass, and the input image. The predicted mass and the target mass don't agree very well for this particular example right now. Partially that's because I cropped it way down and then I didn't resize it. Also, it just didn't run for very many epochs. So these are the kind of things that you kind of need to play around with to better, to suit the needs of the particular problem that you're working on. So that's kind of it for this discussion. Again, I didn't want to get too far into some of the details on the nuances of different UNet implementations. I think that the UNet's a very versatile tool for computer vision, and it's specifically useful for neutron beamlines. And at this point, I'm happy to take any questions.
All right, I'll unmute myself and then I will talk. Thank you very much, John. I'll remind everyone to submit your questions via the Q&A at the bottom of the screen. And we do have a couple of questions that are waiting. So we're going to go ahead and get started with those. So the first question on deck is, can you discuss some of the other applications of the UNet?
Yeah, so I did talk about that a little bit already. Medical applications for sure are one of the bigger ones, and image contouring in like CT scans in particular for trying to do segmentation of organs and cancer and tumors in medical imaging. That's a pretty widely used application of the UNet.
Awesome, thank you very much. Next question up is it possible to accomplish the same thing with a feed-forward network? Not sure what same thing refers to, but.
Yeah, so I think that's an interesting question. Essentially, I think the question is asking, can you do image contouring with a feed-forward network and not use a UNet? Just use a straightforward neural network where you do pixel-to-pixel image contouring. The short answer is yes. Essentially, you have a lot more inputs and a lot more outputs, so it creates a bit more of a complicated structure. The UNet is a convolutional neural network, so it's optimized for processing images using the filters, versus a feed-forward network is just going to kind of brute force, say, map this pixel to that pixel, and it might have trouble identifying particular sort of features that are consistent across the image. But it could probably work. It's just not going to be as efficient.
Okay, thank you. Next question up is, looks like it's a two-part question. Can you explain further the relationship between the number of filters and layers you chose, or is the standard UNet? And then the second question is, how does this change the number of classes in the masks and segmentation? And I will send that to you in chat so you can look at it.
Okay. Yeah, so in terms of like the number of filters and layers, it's not necessarily, I think it gets down to how complicated the data set is. This is like one of these weird things where it's a little bit of an art, less of a science. I think that that's true in a lot of machine learning tools. It's going to be specific to your data set. So you can probably simplify it down as much as possible. Usually I try to make it as simple as possible as a starting point, or as simple where it still works. But for this particular example, I mean, it seems like it's set up to work for this data set, and it does, but is it the most optimized example? I'm not sure. I think that tends to be very data set specific. And I'm not sure if Abdu has a lot of experience working on the UNet for the Topaz data. I don't know if he has any comments on the, like specifically for the Topaz data set, do we need to like play around a lot with the hyperparameters?
I think for the Topaz data set, we fixed the number of filters. I mean, for most of our tests, the images are quite simple compared to, I mean, as you mentioned, medical, compared to medical images or even satellite images. So we didn't do a lot of hyperparameter tests to see how sensitive they are, because I think we used about 6 filters and it did work quite well for HB2A and Topaz.
Usually my experience is that the examples have a lot more filters or a lot more complexity than is necessary for some of the applications that we're doing specifically, that you can really simplify it down to just kind of some basic structures.
Yeah, that's correct. Yeah, one last point. I think the network was more sensitive to the size of the images than anything else. So the Topaz data set are quite large. So I think Topaz was like 1200 by 900. So we had to downsample them to 128 just to be able to have a model very quickly.
All right, thank you. We have another question lined up here. This is, are there any performance trade-offs using RGB versus black and white / grayscale images? And that's from Stephen Webb. Hi, Stephen Webb.
That's a good question. I mean, I think that I don't have a good answer for that right away. It's something that we could look into. I know that for the HB2A and the Topaz, I think they're both RGB, even though they're black and white. But then I think we basically convert them to grayscale, I think, before training. So we're not actually training on RGB. We're just training on a grayscale image.
Awesome, thank you. One more question here. It says, I'm curious about how sensitive the training is to batch size. Did you have any comments on that?
Yeah, so for the batch size, there's a couple of things. Usually my thinking is that to make the batch size as small as possible while not overfitting, that's kind of the goal when optimizing for the batch size. But the batch size, of course, affects how much memory you're using when you're training, because you're trying to load the images. So if the batch size is too large, then it might actually just not be able to train depending on what you're running on. So there's sort of there's some workarounds for that too. But usually at a high level, try to reduce the batch size as much as I can while not sort of affecting the fitting.
All right, thank you very much. It looks like we're just coming up on our 25-minute time, so we'll go ahead and wrap it up. But I want to say thank you to everybody for participating today. Thank you for joining us. Thanks to Dr. Edelen and Dr. Diao for facilitating this webinar. Please watch your email. We'll be sending you a link to the YouTube video recording of this in about a week. And I hope you guys all have a wonderful June, and we'll see you next time.